The tide withdraws from the ancient shoreline, leaving behind a glistening carpet of organic droplets, hundreds of thousands of tiny worlds trembling on the warm mineral surface. Sunlight glitters upon their membranes. A gust of wind passes, almost undetectable, but enough to set entire clusters of protocells quivering like beads of dew. Their fragile walls shimmer with the faintest rainbow sheen, each droplet only a fraction of a millimeter wide, yet bursting with chemical possibility. Within these membranous spheres, life's earliest ambitions awaken. Episode 4, Dance of Molecules, Part 2. One protocell elongates slightly, its membrane thickened by the accumulation of fatty acids. The layer flows, reshapes, stabilizes. The internal soup is not lifeless. It churns with polymer's random chains of nucleotides, mostly unstable, many doomed to break apart in minutes. Yet a few strands endure long enough to influence their survival. These survivors do not yet live, not truly, but they persist, copy, mutate, and interact. And persistence is the first whisper of evolution. A strand of RNA-like polymer coils in the center of a protocell, just stable enough to resist immediate degradation. Its backbone is held together by chemical bonds, more durable than its unruly rivals. Around it drift fragments of proto-ribose, short catalytic sequences born from chance. They bump, collide, fall apart, but sometimes, rarely, they fold into shapes that matter. One fragment curls into a hairpin, bending in the presence of magnesium ions and stabilizing into a primitive catalytic structure. It catalyzes a reaction that allows additional nucleotides to stitch onto the main polymer. The chain grows longer, not elegantly, not efficiently, but measurably. For the first time, a protocell gains a replicative advantage. Nearby, another protocell struggles. Its interior polymers break down faster than they build. Its membrane is too permeable. Ions rush in unregulated heat, destabilizes it. With a soft tremor, the droplet collapses, bursting into the thin tide. Its chemical legacy dissolves into the water. No trace remains. The shoreline is merciless. And so evolution begins its first dance on a stage of stone and water. The protocell with the growing polymer drifts slightly when the tide washes over it. The water's touch is gentle this time, not enough to dislodge it. Instead, the flow brings fresh nucleotides, amphiphiles, mineral ions, and stray catalytic fragments ingredients essential to its chemical persistence. The protocell absorbs some molecules through its semi-permeable membrane. The polymer inside folds and refolds, its shape imperfect but functional enough to continue elongating. Every time the strand folds differently, the protocell's fate shifts. Some folds accelerate reactions, some halt them, some destabilize the entire droplet. The shoreline becomes a battleground of molecular geometry. A second protocell forms nearby, its membrane unusually enriched with slightly longer chain lipids. These longer chains confer stability. Sunlight cannot break them apart as easily. Its membrane is tough, but selectively so, allowing entry of only certain small molecules. Inside this protocell, a different polymer system emerges, shorter but more diverse. Instead of a single principal RNA strand, it hosts multiple small catalytic motifs, a cleavage catalyst, a primitive ligase, and two random coils with no apparent function yet. the cleavage catalyst is unstable. It breaks almost as quickly as it forms. But during one brief, coherent state, it severs a longer random polymer and creates a new sequence 1 with folding potential. This accidental fragment becomes a molecular pioneer. When folded, it forms a loop with a catalytic pocket that can join nucleotides end-to-end far more efficiently than its ancestors. The protocell becomes a hotspot of polymerization. Its interior begins to fill with new catalytic species. For a moment, it teeters on the edge of chemical chaos. Then stability emerges from the turbulence. A lipid vesicle adjacent to it collapses from osmotic shock to many ions rushing in. But the resulting burst splashes fresh lipids across the surface of the catalytic protocell. They merge, strengthening its membrane further. It grows slightly, expanding its volume. With expansion comes dilution. Some catalysts become too sparse to function, but others remain just concentrated enough to continue their roles. The protocell becomes a compromise between growth and efficiency of balance required for survival. On the opposite side of the tidal pool, a cluster of protocells forms in the shelter of a mineral overhang. These droplets contain a different chemistry entirely. Instead of RN, A-like polymers dominating their internal environment, these protocells rely on peptide-like chain-short amino acid sequences formed during storm discharges in the ocean. These protopeptides behave differently from nucleotides. They fold quickly, forming rudimentary structures, loops, sheets, coils. Some possess weak catalytic activity. One peptide acts as a scaffold, stabilizing other molecules so that reactions occur more predictably. It anchors itself into the membrane, becoming an early membrane-associated catalyst. Another peptide behaves like a primitive transporter. Its folded surface contains a channel-like depression that attracts specific ions. The peptide does not intentionally move ions, but its structure nudges them into the protocell interior. With each influx, chemical diversity inside the droplet increases. A peptide-rich protocell becomes a distinct lineage, drifting in the cradle of tidal cycles. The sun reaches its zenith, bathing the pool in relentless light. UV radiation slices through the water. Many polymers break apart. Some protocells rupture, but a few have evolved thicker, more ordered membranes. These membranes contain patches enriched with isoprenoid-like lipids, precursors to the molecules that will one day define arsenic-tolerant expression. These lipids are resistant to UV-induced fragmentation. They act as primitive sunscreen. The pool's surface becomes a shimmering mosaic of shimmering droplets, some bright, some darkened by pigment-like molecules accidentally synthesized inside. These pigments absorb destructive radiation, converting it into harmless heat. Their presence is a fluke, a byproduct of unrelated reactions, yet it grants survival. When a peptide inside one protocell binds to a pigment, the combination produces a rudimentary light harvesting cycle. Not photosynthesis, not even close, but the faint hint of regulated photochemistry. The protocell with this pigment peptide complex persists through the harshest midday flare. Evolution is blind, but chemistry remembers. As the sun descends and shadows lengthen, the pool cools. The tide returns slowly, swirling the protocells around one another. Some droplets fuse. Fusion is dangerous. When two protocells merge, chaos erupts. Two chemical systems collide. often fatally. Most hybrid droplets destabilize within minutes, but rarely, perhaps in 1 out of thousands of fusions, the merged protocell inherits stability from both parents. A polymer from one lineage finds itself suddenly bathed in monomers from the other. A peptide catalyst encounters a new substrate. A membrane from 2 different chemistries becomes more robust. These rare survivors represent the earliest molecular species, lineages indistinguishable to the naked eye, but profoundly different in their internal logic. A fused protocell slowly drifts toward the shallow edge of the pool where warm mineral surfaces offer protection. Inside, its polymers begin performing reactions they were never designed for. A ligase-like ribozyme encounters a peptide scaffold and folds around it, using the peptide as support for more efficient catachesis. Reaction rates increase. Polymer sequences lengthen. Mutation rates rise as thermal cycles drive denaturation and refolding. The droplet becomes an engine of chemical innovation. Across the pool, a dark protocell forms in the shadow of a rock. Its membrane is rich in polyunsaturated lipids, unusual for this time, but born from accidental synthesis involving sunlight, iron ions, and simple aldehydes. These unsaturated bonds create fluidity. The membrane ripples, its flexibility allowing the protocell to engulf small organic clusters. The process is primitive phagocytosis, like absorption, not intentional, but driven by surface tension dynamics. As the droplet engulfs these clusters, it gains access to concentrated organic precursors. Its internal chemistry flourishes. For a moment, it seems invincible. But the same fluidity that grants advantage becomes a vulnerability. An abrupt temperature shift caused by a passing cloud and rapid cooling triggers membrane instability. The droplet stretches, thins, and ruptures. Its contents spill across the mineral surface. The chemical lineage ends in seconds, erased from the evolutionary record. The pool's history is written in extinctions. But amid loss, a new protocell lineage rises. Its membrane incorporates both saturated and unsaturated lipids in alternating patches. This mosaic pattern grants it an unprecedented stability across temperature fluctuations. Inside this droplet, a self-templating RNA-like polymer finally emerges, long enough to hold structural motifs that enable catalytic pockets to form consistently. Its replication is slow, error-prone, and messy, but it is replication nonetheless. A crude cycle emerges. The strand folds, catalysts bind, monomers attach, errors accumulate, variants form. Some variants fold better. Chemical selection becomes Darwinian selection. Nearby, another lineage relies not on nucleic acids, but on protometabolic networks. Iron-sulfur clusters form spontaneously along membrane surfaces. These clusters act as electron carriers, facilitating redox reactions. A droplet-rich infest clusters stabilizes into a microreactor. Redox cycles inside it drive synthesis of short peptides. The peptides interact with the mineral clusters, forming primitive complexes reminiscent of ancient metabolic enzymes. The protocell becomes the seed of what will one day evolve into the citric acid cycle. Glycolysis and respiration mechanisms, billions of years in the making, yet their shadows flicker faintly here. The tide recedes again, leaving protocells stranded on the warm rock. Those with robust membranes survive desiccation cycles. Dehydration concentrates their contents, accelerating polymerization reactions. Those with fragile membranes crack open, but even their demise seeds the environment with new molecules for other protocells to absorb when the tide returns. Cycles sculpt life more effectively than intention. As midnight approaches, the pool darkens. The only illumination comes from faint chemical luminescence, sporadic flashes caused by redox reactions, metal-ion collisions, and the spontaneous decay of unstable molecules. In the darkness, protocells drift, merge, divide irregularly, and reshape into new forms. Some droplets split due to mechanical stress, producing daughter vesicles containing only fragments of the parent's chemistry. Some daughters inherit critical catalysts, others inherit none. Differential survival begins to sculpt the protocell population. A lineage of droplets emerges with a particularly fortuitous combination. A hairpin ribozyme capable of template-directed synthesis, a membrane-bound peptide transporter, and a pigment-like molecule capable of dissipating harmful UV radiation. Though crude, these three molecular species form a synergistic network. When the tide returns at dawn, this lineage remains abundant while others vanish. Chemical alliances become evolutionary strategies. As dawn breaks, the first rays of light dance across the pool once more. New polymers form. Old ones decay. Protocells continue their struggle in endless cycles of fusion, rupture, replication, and transformation. Across thousands of generations, lasting mere minutes, the chemical networks inside droplets refine themselves through brutal filtering of instability. A droplet in the center of the pool becomes the first protocell capable of quasi-division. It grows as lipids accumulate on its membrane. Thermal expansion stretches it. A constriction forms in the middle. The membrane wavers, thins, and eventually pinches into two parts. The daughters are unequal. One inherits more polymers, the other fewer, yet both survive. Imperfect reproduction marks the dawn of lineages that extend beyond single droplets. The pool becomes noisy with chemical ancestry. Lines rise and fall. Species emerge and collapse. Molecules compete, cooperate, merge, and mutate. Every failure reshapes the chemical landscape. Every success echoes across generations. By sunset, one lineage dominates. Not because it is superior, but because its molecular toolkit happens to withstand the relentless cycles of light, heat, ions, and tidal motion. This lineage's internal chemistry is stable enough to endure, yet flexible enough to innovate. It hosts ribozymes that copy themselves, peptides that stabilize membranes, pigments that safeguard reactions, Fes clusters that guide protometabolism. Each molecular species is still crude, primitive, inefficient, but together they form the first faint outline of what we will one day call a cell. When the final tide of the day rolls in, the protocell cluster drifts as a trembling patch of life's earliest ambition, held together only by the fragile boundaries of lipid films and the determination of molecules struggling to outlast chaos. And deep within one droplet, a polymer folds into a shape never seen before, catalytic pocket, capable of stitching nucleotides with unprecedented accuracy. A quiet shift occurs, not dramatic, not visible, but decisive. The dance of molecules has entered a new phase. Evolution has found its rhythm, and the age of protocells edges closer to the dawn of true biology. Next episode, 5 Survival's Edge. This is HistoPrimordia, where history comes alive. Thanks for watching. Subscribe now and join us for the next episode story.